All right, thank you very much. Great to be here. What I'm going to talk about this afternoon is the use of transcranial magnetic stimulation, also called TMS for short, to enhance psi using mind-matter interactions as a model. So I just want to mention this is a collaborative effort. So I've got collaborators, Malcolm Binns, Jed Meltzer, Rahila Hashimi, and Robert Chen. So let me just advance here. What I'm going to do is I'm going to give you my key messages right at the beginning, and then I'm going to give them to you again at the end, okay? So the key messages are that reversible TMS-induced lesions, and it's very important, they're reversible, and we're reducing function. TMS can do both, increase or decrease. But TMS-induced lesions, which is what I'm calling them, in the left medial middle frontal brain region, up here medially on the left, significantly enhances psi effects as measured by mind-matter interactions. We've got a p-value of 0.006 and effect size of 0.38. This study represents the third replication of this suggested finding. It's actually 4 times. The first time is not a replication, but it's 4 times we found it. And individuals, and a real message is that individuals with reversible lesions from TMS, they could comprise an enhanced sample for studying all kinds of psi. And also, the psi effects, they're very small and hard to reproduce. And the main message is there's a reason for that. And it's because the brain blocks psi. And what we're doing with TMS is we're unblocking it. So what I'm going to do is I'm going to review the rationale for why we even talk about the brain on understanding the mechanisms of psi. And also I'm going to present the new data from our TMS study in 108 participants showing what we found. So the question is, why focus on the brain? A lot of people aren't interested in that. They think the brain is something off to the side from the things that you're describing. Look, the brain is part of the equation for psi. There's no psi without the brain because you can't make an observation of the psi. So the brain is relevant, okay? Identifying the mechanisms of the brain in the brain mediating psi, first of all, it can lead to a whole set of new methodologies, which is what I'm talking about. Can lead to practical applications, which I'll talk about. And I'll tell you, if you want psi, what you're doing to be, and what we're doing, to be accepted by mainstream science, you need to include the brain. Otherwise, it will not happen. So I just want to go back into what got me started into all of this. Now, Henri Bergson, in the early 1900s, talked about the brain as a filter to inhibit psi. And he actually said there's a reason for this, because we would be flooded by information overload, and we wouldn't be able to survive because we'd be so overloaded. And he said that it's actually deficits in this filter that leads to the emergence of psi. And I believe that when people are doing this and they have psi effects, they're actually, in a healthy way, suppressing a part of the brain, a part of brain function. Now, what Bergson didn't do is he didn't localize the filter to the frontal lobes, which is what my group is doing. And the reason for postulating the frontal lobes is if you look at psi-enhancing states, they're often, meditation, hypnosis, they're often associated with reduced self-awareness. Now, the frontal lobes, one of their jobs is to increase self-awareness. So the idea that I got was to reduce frontal function in order to simulate the psi-enhancing state of reduced self-awareness. That's the rationale. So the initial research, and I presented it here before, but I just want to go over it. What we did is I studied patients with brain damage. And we used from the pair group, the random event generator, portable random event generator. What this does, it gives you zeros and ones with an equal probability. That tells me what I got left. Okay. All right. Anyway, there's 200 bits comprising a trial. Basically, you get a number that is expected to be 100. And you try to think high numbers and low numbers, okay? Now, the thing that we did is we translated the numerical output into movement of an arrow on a screen, because people with brain damage have great trouble thinking high numbers, low numbers, but you show them an arrow and you say the arrow's pointed to the right, get it over there, they can do that. So high numbers, if the intention was get high numbers, the arrow pointed to the right. Low numbers, the arrow pointed to the left. And this is what they saw. So the one, if we're asked people to point to do, making the high numbers, the arrow going to the right, they saw the image on the left. Otherwise, they saw this image. They only saw one of them. But their task was get that arrow over to the side, okay? So here is a slide. It's not working, but I was... What's that?
If you click on it, it might go.
Click on it.
Or it may go to the
It's not going. Anyway, that arrow jumps around depending upon what you're thinking about. Okay, and what we did that's different from the pair group, they didn't have a control group. What we did is we had, when patients finished, these were patients, when they finished, they left the room and the random event generator ran by itself and collected data without a patient in the room. And our comparison was what the random event generator, the reg, was doing when the patients were in the room versus when they weren't in the room. And we had two patients, one with a focal lesion in the left side and another one with frontal temporal dementia. And we looked at the lesion overlap. And the lesion overlap was in the left medial frontal region involving Brodmann areas 9, 10, and 32. And where it is, it's right here. This is the middle side of the frontal lobes, and it's right in this area here, this reddish area. That's a region that inhibits psi. So what we did is we got a grant. I mean, I'm not going to get 100 patients with that kind of lesion, but I can get 100 normal people. So we got 108. And we used TMS to induce reduced function in that area, okay? It's temporary. And we made an a priori hypothesis. This is very important because we've actually submitted this to a mainstream journal. And we said that healthy participants with reversible lesions in the left medial middle frontal region will show larger right intention effects because it was only getting the arrow to the right side, opposite to the side of the lesion, which makes anatomical sense. And they would have a greater effect than participants without a lesion. And that was in our grant before we did the study. So it was an a priori hypothesis. We didn't make it after we started the research. And we used what's called theta burst stimulation. This is a TMS protocol, and it has an effect lasting about 20 to 30 minutes. And this is the way we designed it, okay? So we randomized our patients. There were 36 who had TMS giving a lesion where we predicted it would have an effect. We had 36 who had a lesion put on the other side, and we didn't predict that anything would happen there. And then we had 36 who got stimulation, but it was sham. So there is stimulation, but there's no actual effect on the brain, okay? And we predicted that we'd only see an effect in that group. So participants were in a soundproof room. They were asked to move the arrow to the right for 500 trials, to the left for 500 trials. It was counterbalanced, like half go to the right first, half go to the left. And we had 1,000 control trials after they left, because they did 500 trying to get it to the right, 500 trying to get it to the left, and we had 1,000 control trials. And this just shows you that when we asked them to do it to the left, that's what they saw. when we asked them to do it to the right. That's what they saw. This is think low numbers. This is think high numbers, but it was all transformed to an arrow. Okay? Now, this whole thing should take, but they get the TMS, then they got to do the study. And so that takes about 23 minutes. So it wears off. So what we did is we did an analysis assuming there was no wearing off and there was nothing significant. But then what we did, and this is standard, because we weren't looking for the duration of the effect, we were looking to see if there was an effect. So we used a weighting where the earlier trials are given more weight because it's closer in time to the TMS. So this is a weighting procedure applied to reduce the influence of observations collected after the potential wearing off. And we found, when you did this, highly significant, 0.006 with an effect size of 0.38. Now this slide has all of the data. It's very complicated, but what we're looking for is very simple. So what I want to show you here, I'm going to explain it, and then I'm going to show you the results. But what this slide shows, on the bottom are the direction. First of all, on the top is the left. This box is the first 500 trials. This is the second. So the greatest effect of TMS is on the box on the left. Now, on the bottom, here's left TMS, and this is trying to get the arrow to the right, trying to get to the arrow to the left. This is right TMS, arrow to the right, to the left, and this is sham here. The red is the intention, trying to do something. The gray is the control trials. So we're comparing the red to the gray. This is the average reg output, and the expected is 100, which is this line. Okay? Now this is the finding. There are lots of comparisons you can do, but if you make a hypothesis, you've got to test only your hypothesis. Otherwise, you're fishing. So look at this. These are the left-sided lesions. This is the intention, trying to get the arrow to the right, and this is the control. Here are the sham. And the comparison is this. We take the patients with the left TMS and we compare the performance here to the performance here, that difference, and we see if it's bigger than the difference in the controls. And it's a whopping difference. Now, if you look at what's happening, this is the expected value. Almost the vast majority of patients, of participants, had the arrow going to the right. And this is the median, and you can see it's very symmetrical, whereas in all the other comparisons, there are some that are above the line, some below the line. Whereas what this means is that the vast majority of people, when they were asked to move the arrow to the right, did move the arrow to the right. But when you look at the other groups, it's all over the place. So this is it's called an interaction. So what you're looking at is here are the move the arrow to the right. This is, the red is trying to do it, the gray is the controls, and you see if they're different, okay? But it's not enough to just see if they're different. You have to see if they're more different than in the sham, okay? So the difference is greater in the group of interest than in the sham. And that's called an interaction effect. That's the only comparison that counts. This, if you want to look at it simplistically, all it says is when you try to move the arrow to the right, it's better than controls. But remember, you've got to compare it to the other. This one I'm going to skip over. This one is showing you what happened for the right-sided stimulation. And these are you can see that's the median, okay? And you can see that nothing is happening. It's all hovering around 100. So there's no effect when you stimulate the right. And I'll give you the reason for that in a moment. I got 5 minutes left, I'll be able to do it. Okay? Does everybody understand what I just said? Because it's, you know, if you're not familiar with statistical analysis, this can be very confusing. Okay? So that's it. I'm going to give you the key messages again. That reversible lesions in the left medial middle frontal region significantly enhance psi effects, okay, as measured by the mind-matter interactions. This is micro-PK. Now, we haven't done it with other types of psi. This is the third replication. There's no decline effect here. This is a strong effect. And these, when you use TMS, you can get a group where you can have an enriched sample. So if you're doing healing, a lot of people are interested in healing. What you need to do, this is a hypothesis, haven't done it, okay? But what I think you need to do is you need to give TMS to the healer. Not whether you give it to the person being healed or not, I don't know whether that'll work. But you give it to the healer because it's the healer that needs to unblock the psi effect, okay? So this can, you take all your experiments, redo them with TMS. And this tells you why, you know, because we're studying patients where the brain is blocking the effect. And that's why the effects are so small. And that's why it's so hard to replicate. But we've replicated it 3 times. So I think this is very striking. I'm hoping the mainstream journal will accept it. What they asked for is revisions, which actually is a very good thing when a journal asks for revisions. But they were tough. I got 3 minutes. Okay, I want to have some questions. This. Oh, I got two more minutes. Okay, so I wanted to present this here. I presented my first work here, published my first paper in JSE. So I think that this can be a game changer. You know, I know that, you know, the brain doesn't figure in for a lot of people, but it does figure in because the brain is getting, I'm a neurologist, okay? And the brain with psi is actually getting in the way. And it's getting in the way for very good reason. Because if we didn't suppress it, our world would look very different than it does now. And it may not, in fact, our world wouldn't have evolved to the way it is now. We evolved to be private. We evolved not to be sharing everything. We evolved not to be, you know, doing macro PK and micro PK. That's our society. And what we are finding is that there's this signal that something is happening. And we're trying to show it. Well, if you want to show it, you got to unblock the inhibitor in the person who's doing it. Because all of these experiments involve people. Or they involve plants, they involve, but there's a person somewhere in there that's making the observation. And the brain is limiting that. So that's my message. The brain is actually very important. Now, we want to do this. TMS you can't do at home. You can do it at home, but not this kind of TMS, okay? But there's TDCS, which is electrical. Now that, there are home devices. So my next study, if I can get some funding, is going to be to do this with TDCS. Because that can actually, you can do that. I mean, I'm not in favor of people doing a lot of these kinds of things on their own. It really, you need education, you need training. But at least it can be done outside a laboratory. What I just did, you can't do on your own. It's just too complicated. So I'm going to stop there. Thank you. Happy to answer questions.
Yes.
Lawrence, your research is very impressive, and I'm glad to see that you're doing research on methods to enhance psi abilities. And the question I was going to ask you, you partially answered at the end, and that is, of course, many people would be and healers would be skeptical about using some methodology that, even if it's reversible, lesions, and maybe it would seem to be reversible 100%, but they would still be skeptical from a psychological standpoint. So my question was that, are you looking into methods of enhancing psi abilities that would not require reversible lesions, like you suggested, and maybe were either direct current stimulation, which you mentioned, or pulsed electromagnetic fields, or some other methodology, such as Michael Persinger made years ago with the God helmet, that kind of thing?
Okay, first of all, I mean, TMS is really a relatively safe, they use it clinically. They use it for depression, for OCD, so it is, you have to be careful, because there's certain drugs that could produce adverse events. Seizures can occur, they're really quite rare. But whether it's TDCS or TMS, or if you're doing any kind of stimulation, you're doing something to the brain. You can use TMS to enhance. So we shouldn't be caught up with the idea of inhibiting versus enhancing. You're doing something to the brain. Can you do this without any of this? Yes, you can. I mean, people who seem to have abilities, psi abilities, are, I think, shutting off their medial frontal lobe. But that takes a lot of work. And you can see how hard it is to replicate that kind of thing. So the answer is, psilocybin, for example. You know that psilocybin reduces medial frontal blood flow. And if it has any effect inside, that's probably the mechanism.
So your work, I think, really advances our psi to a new level by connecting it to brain activity. And you've been doing this now for a while, and it's great. I have a question, and that is that I'm really concerned about the experimenter effect here, because you're the lead investigator. This is following your intention, and you're getting a significant psi effect in a random process. We and many others have seen huge psi effects like this. Do you have a way to test for that?
Yeah, I'll tell you exactly why this is a psi effect. Because I started this work in early 2000s. So I've gone, I'm telling you why it isn't any kind of effect here. I've gone through 2 different rigs. When I set up these experiments, I plan it. I have my research assistants do it. I have nothing to do with it. I don't even know when the patients are being tested. So this has gone through several research assistants. When the MRIs are analyzed, that's done in a separate lab, has nothing to do with me. The statistics I don't do. I have a statistician doing it. I just set it in motion and then go about my business. So I'm actually far removed from it. What I do is I design the study. So could it be an experimenter effect because I'm there? If it is, I'm telling you, there's so many people involved in this that, in fact, one of the investigators was just very negative about this. One of the TMS people, 2 TMS people, one was very negative, and he sort of agreed to do it because he was me, and he was floored by the fact that it came out to be a priori hypothesis. So what about his effect? So I don't think it's an experimenter effect. And, yes, I'm the constant, but the effects are too strong.
Just a quick add-on. Does somebody else complete the statistics so they see the result before you've ever seen it?
Yes, our statistician does. I couldn't do this. I'm not a statistician. You realize the complexity of this and the discussions that went with one of the neuroscientists. And so I did the statistics for the first one because it was very simple, the t-test. And I did it for the second because it was a t-test. This is not a t-test. This is so complicated. It's way above my pay grade.
One more question.
Morris, first of all, I want to say congratulations. I first saw you present on this in 2016, your first pioneering work in this area, and just to see you kind of build on a body of research over a number of years and to get to this point is just really fantastic. It's a credit to the work you're doing. Beyond
Thank you.
the neurology aspect, I would ask you to speculate in terms of like mental process and how these might be related. So do you think that your effect could be somewhat attributable to like a dampening of the monitoring processes of the anterior cingulate? So, people are processing feedback very differently than they would, without that kind of TMS intervention. Does that make
Well,
sense?
It could be. What I think is this, and it has to be tested. I think that psi requires reduced self-awareness, and reduced focus on oneself with very good attention. That's my hypothesis. That's one of the things I need to now do, is look at it in relation to that. I don't really know what the psychological mechanism is, but we never saw it in the right frontal. And I think the reason is that the right hemisphere is very important for attention. And I think that when people have lesions on the right side, they're not focusing as well. So you need that unique thing about reduced self-awareness and good focus. And that may be what happens in some forms of meditation. So I don't know. That's something that we need to study. But now that we've got this effect, replicated 3 times, and I get funding, I'll start to go on to these other things.
Thank you. Let's all thank Lawrence.